I'll start right off with sort of why we got interested or why we're interested, why we like covalent modifying molecules. Well, we study proteases in particular. And over the years, we've been working on these classes of covalent inhibitors of proteases. One class of enzymes we work on are called the cathepsins. These are lysosomal cysteine proteases, so they have an active site thiol. And we design molecules that look like a substrate that bind in the active site of the protease. The covalent bond forms initially by attacking the carbonyl and then moving over to form a very stable thioether linkage. And when that happens, it releases part of the molecule. And if you design these molecules properly, you can get this sort of quench configuration where you have molecule in solution here, unbound, has a fluorophore, but it's quenched by a nearby quencher. Here's the protease with its active site, and the yellow is the cysteine. It goes into that active site like a substrate, that covalent bond forms, and now you lose part of your molecule and it turns on a fluorescent signal. So you now have a way of tagging the protease. So it's a way of visualizing where the active protease is in the context of either a cell or even whole organism. And we've been applying that in actually all the way up into whole animals here, where you can introduce one of these covalent probes that turns on when it sees a protease, and it turns out these lysosomal proteases are highly abundant in tumors. So if you inject one of these probes into a mouse bearing tumors, you can see the tumors start to become fluorescent as the proteases activate the probe. And so this is useful for image-guided surgery, and a version of this molecule now is in clinical trials for actual translation into human use. The interesting thing is because it's covalent, we can, after we're done imaging a live animal, we can remove tissue, the tissue's fluorescent, and we can actually visualize by SDS Phage gel what got labeled. In this case, you can see multiple bands. These are individual lysosomal cysteine proteases that got hit by the probe. And we can see it because they're now permanently tagged with our fluorophore, and we can just scan the gel. So this is really cool. We've been studying this for a long time. Part of the issue, though, is that we tend to make these molecules that are very short, like linear peptide-like sequences. In this case, you can see it's just a dipeptide with a sort of reactive warhead attached to it. So it's hard to get selectivity. You get things that bind to multiple targets. Here you're hitting multiple cathepsins. So if you really wanted to target just one of these cathepsins, you probably need something a little more significant than this linear peptide. And so one of the things that got my attention was actually work that was done by Christian Heines's group and published a number of years ago. But essentially, rather than using a linear peptide to bind to, like here again is the lysosomal cysteine protease, Christian's group developed these ways to make these constrained peptides or bicyclic peptides. So these are much larger peptide sequences, many more amino acids in them, and then they have a red core chemical group that constrains it into this bicyclic structure. And the result of that is you end up with a globular, more protein-like fold of your peptide, and then you can interact with a much larger surface. You can see here compared to the molecules we're developing. So this got my attention because when Christian published this initial paper, the way they get at these molecules is that they're, obviously chemically very difficult to make, billions of sequences here that you could then test for binding. So what they use is they use a phage display approach. So the idea is let the phage make the peptides, and you can build billions of peptide sequences on the surface of the phage. You come in with your chemical modifier right on that, right on the phage particle itself, and you can actually form these constrained bicyclic structures on the phage particle, and then you screen and you can identify bicyclic peptides right off of that without having to chemically synthesize so many. And so what was really cool is in some of the first papers back in 2012, sorry, actually it was an earlier paper that Christian published. What really got my attention was that the target here was a protease called UPA, the human UPA, and you could see they could use phage and they got this optimized binder. What was really neat is that if you look at the mouse version, which is highly homologous, in fact, almost identical in sequence in a lot of ways, you get 3 orders of magnitude shift in terms of potency. So you're getting an incredible level of selectivity against other proteases. And that's simply because you've now created something that looks like kind of a small protein-protein interface. So we started to think about this. Well, can we use this for our covalent drugs? And so actually I went to Christian's lab back in 2012 for sabbatical. He's at EPFO in Switzerland. I had this nice view of the lake here from Christian's lab. And I worked with this guy here, Xu Chen, who was a student in his lab at the time. Got in the lab and did some work myself, which was really fun. And then eventually brought that work back to my lab. And Xu finished his PhD and then came to my group as a postdoc. And then another postdoc, Scott Lovell, joined. But basically, we took the same idea of the phage expressing a peptide with, in this case, 2 thiols, 2 cysteine residues. We then created these warheads that reacted with the 2 thiols and that also contained a reactive electrophile. So now we have the cyclic peptide constrained part that we're interested in for for mediating binding, but we also now have the covalent part that we want to tether these things in. And now you make libraries like this where you can put whatever warhead you want on your phage, and you can do your iterative panning against a target like a serine or cysteine protease is what we've been doing. You go through these cycles, and now the big difference is these covalent phage should stick and be permanently stuck, so you can use extremely harsh washing conditions to get rid of all the phage you don't want that are just reversibly bound. And then at the end, you actually cut your protein off of your affinity resin, and that elutes the phage, and you can then optimize various cyclic peptides that contain a reactive warhead. And you can see here 2 examples of vinyl sulfone for a cysteine protease and a diphenyl phosphonate for a serine protease. So one of the first targets we did was TAV protease. I'm going to kind of go through this a little quickly because this is published data, and you can go look at the paper that came out a couple of years ago. But we chose TAV because it's a cysteine protease and it has a very defined primary sequence specificity. So we wanted to see if we could make cyclic peptides that would bind with that same kind of specificity as this linear peptides. So we made phage that had a number of variable positions between 2 cysteines, and we used a vinyl sulfone attached. We cyclize on the surface of the phage and we do our panning. And after several rounds of panning, we got a sequence that came out that was around 5 micromolar binder and not super potent. But what was interesting is these colored residues are known consensus binding sequences for TAV. So we were getting the right kinds of sequences, but they were in a very different orientation than what you would get on just a straight linear peptide. We then did some alanine scanning where you kind of walk through these positions and then test potency. And you can see these positions here, in particular, these 3, the E, L, and Y, are really important because if you mutate them, you get a big jump or loss of potency. However, if you make mutations on the other end down here to alanine, we didn't see really much change. These are all like low micromolar inhibitors. So it told us that we could further modify these positions. And ultimately, using some basically peptide medchem, we were able to get to a sequence that was optimized, that was a sub-micromolar binder for TAV. And so that was pretty exciting. So it's potent, right? Here it is binding to TAV. This is the cyclic peptide down here containing the vinyl sulfone. So it's less than 1 micromolar. But what was really interesting was the specificity. So to show the specificity, we made the linear version of the optimized peptide. So this is the optimal linear sequence that the protease likes. And we put the same warhead on, the vinyl sulfone. When you put that on, you get a TAV protease inhibitor that's about 2 micromolar. So it's similar to what we got with the cyclic peptide. But when you then test it against other cysteine proteases like cathepsins, you can see it's not a great inhibitor of CAT-S, but it's an incredibly potent inhibitor of cathepsin L, which means this is the problem with the linear sequence, right? You got a good optimized sequence for TAV, but this other enzyme likes that same sequence pretty well. The big difference was if you can now look at our cyclic peptide and test against those 2 cathepsins, you see absolutely 0 activity. And that's simply because this is such a constrained, specific binding sequence for TAV that it just doesn't touch the other enzymes. So that was the kind of specificity we were looking for. And that was one of the reasons why we really wanted to go to these kind of constrained peptide sequences. Okay, so we did some modeling. This is just this is basically a molecular modeling where we tether in the molecule and then look for low energy conformations. And what we can see in orange is the native linear sequence that binds, and you can see these 3 anchor residues for TAV. And you can see in purple is our cyclic peptide, and it nicely overlays with the linear peptide, and it displays peptide residues into the pockets like as if it was linear. But you can see we're now getting all these additional secondary interactions that happen simply because we now have selected using billions of sequences of phage. Okay, so yeah, that was interesting, but TAV is not a super interesting target. So we started to think about other targets. And one of the areas that my lab is interested in is infectious disease, in particular Staph aureus we're interested in. The reason why we're interested in staph is because it's a bacteria that tends to hang out on your skin and it's fine, but once it gets into your bloodstream and you get a systemic infection, it can cause conditions that are very problematic. In particular, endocarditis is one where this bacteria can actually form a biofilm on the valve of the heart. And the problem is that it not only is it a problem for this, it's also a surgical site implant issue. The problem is that you can tell that someone has bacteria, so you can, for diagnostic, you can take a blood culture and say, all right, you have a systemic infection with staph, but it's really hard to say where is that infection. And it's even more difficult to ask the question, are you responding to antibiotic treatment? So typically you have to go in and do invasive things like take out valves of the heart. And what would be really nice is to come up with non-invasive ways to see where the biofilm is present. So one of the things we'd like to do is to develop probes that covalently stick to markers on the surface of the staph, and then those would then annotate where the staph biofilm is present. And that would allow you to do, if it was non-invasive, you could do serial imaging to determine whether you're responding to antibiotics and potentially do things prior to having to go in and do very invasive things. Anyway, so one of the things we did was to take one of these kind of covalent probes. This is a probe developed actually by Ben Cravath's group at Scripps, and it's basically a broad spectrum serine hydrolase probe. So it targets serine proteases and other esterases and lipases that use serine as a nucleophile. So we took this, put it onto the staph biofilm, and we got a bunch of labeled bands, much like I showed you with the cathepsins. But this was a much more diverse pool of things that we were labeling with our fluorescent probe. We were able to pull all those targets out and identify them. We found a whole new family of what we call the FPH proteins, or fluorophosphonate binding hydrolases. And these are all membrane. We think most of them are externally membrane exposed serine hydrolases that contain these red serine hydrolase domains. We don't know exactly what they're doing. I'm not going to get into the biology. We think they're scavenging lipids from the host side. They're almost certainly, most of these are lipid esterases, but they're interesting biomarkers, right? And we can identify them now in the gel. And so we took this one FPHF, which is one of the more abundant ones that expressed really well recombinantly. You can see here, it doesn't have any kind of transmembrane domain, so it expressed very well. And we ran it through our phage panning, and we were able to get cyclic peptides out that bind. All we did was change the warhead here. Instead of using a vinyl sulfone, we used a diphenyl phosphonate here, and we got this is the target one that we got, optimized peptide. Here's a scrambled version. And what you can see is that, in fact, we got a submicromolar binder of FPHF. And you can see when we scramble the sequence, we get a much weaker or almost no significant binding. If we then test it against other proteases like trypsin and then a highly related FPHB that has high homology to FPHF, you can see we get no activity there. So again, we're getting this highly selective binder by having this cyclic peptide constrained backbone. So that's great. So let's talk about some of the more recent targets, because that stuff has all been published, but we're obviously interested in viral targets as well. And I know this is a viral audience. So we started thinking about spike protein. Could we apply this approach to something that's not a catalytic enzyme, but that we want to get covalent binding to a specific protein? And spike is obviously an interesting one, because if we can covalently stick to the trimer of the spike, we can block entry. And there was a paper that came out from Lei Wang's group at UCSF that was really quite interesting, where they took electrophiles, in this case, fluorosulfonate electrophiles, which are extremely weak modifiers of several residues, in particular histidine, tyrosine, and lysine residues on proteins. What they did is they took a molecule that was already a nanobody. It was a small binder of spike protein, and they engineered into it a site non-naturally that encompasses this electrophile. And they could show that now when the nanobody binds to spike, they actually got a covalent interaction to spike. So it's an irreversible binder, and that blocks the ability of that obviously virus to enter. So you can look at actually blocking viral entry this way. So it's basically like using the antibody, but now souping it up by making it a covalent version. So that got our attention, because it suggested that maybe instead of a nanobody here, we could actually just use simple cyclic peptides that we could identify through phage display using these kinds of electrophiles. So that's what we did. We basically took our phage, and this time we used a linker that contained this kind of fluorosulfate, which we knew was, we knew that this would be, potentially a reactive group, and has been shown by Lei's group. So we essentially did exactly what I showed you for the other targets. Again, cysteines with a loop region that was variable. And then we're looking for secondary modifications. So this is a little more challenging, because now there's potentially a lot of spots on the spike where you can get modification. The nice thing with a protease or a lipase target is that you have this highly nucleophilic active site residue. And here we're just hoping to find a passive nucleophile that's going to react simply by proximity. So the way to do this, and it turns out you kind of have to come up with an elaborate panning strategy, because what happens is if you just take your phage library and you pan it on your target, in this case, the biotinylated spike protein, you're going to pull out phage that bind, ideally covalently if you use stringent washing. But you're going to find ones that bind in this, say, pocket here that you're interested in. This would be the ACE2 binding pocket that you're trying to block. But they also bind probably in other locations around on the target. And so you want to figure out what are the critical ones here, the ACE2 interacting ones. And so what we did was to run another series where we used the covalent nanobody that Lei Wang's group gave us, and we pre-block the target. So that should specifically cover the ACE2 binding pocket, and then we do our panning. And so now when we pull out phage on this side of the negative selection, These are ones we don't want, so we can sort of filter them out of the data. So this is how the data looks. We've come up with a data pipeline. At the end of your phage panning, you do deep sequencing, and you identify all the sequences that you pulled out. And what you can do is, compare your enriched, this is the total enriched versus the beads, and then you do the same thing with the negative selection. So this is the positive selection, negative selection. And then you compare the positive to negative, and you're looking for hits down here that are specifically enriched in your positive selection and not in your negative selection. And you can end up with a relatively small number of hits this way. And that allows you to now prioritize those for further development. And if we looked at the ones that were prioritized as being, binders at that interface, the ACE2, we got several clusters here, cluster 1, 2, and 3, and nicely they have highly homologous sequences, which suggested this is probably a real set of binders. And above it, what you're looking at in green are the phage titer, or the number of counts of that peptide at different rounds of panning. And you can see the green shows that we're getting increase, and the red shows that as soon as we switched on round 3 to negative selection, that stopped enriching. So these are the kinds of sequences and patterns that we're looking for. So we took some of those hits, and we then analyzed using an ELISA-based strategy. The nice thing about working on spike protein is there's obviously a lot of interest in assays to measure antibody binding to spike. And so what we can do is just use one of these plates where you have your various spikes, and these are all different spike proteins from different variants of the virus. And you're basically just coming in with an antibody that that's going to bind at that, actually using a human ACE2 protein that's got a label on it. And so you're just measuring the interaction between ACE2 and the target. And if your molecule blocks this, you're going to see loss of signal. So it's simple ELISA in a plate-based assay. And we could then run our top hits. You can see we synthesized about 30 or 40, almost 40 hits from that initial screening. And we did time-dependent ELISA binding assays. And we're looking for things like at the top here, where as we increase the amount of pre-incubation time, we see more and more block of that ACE2 binding to the spike protein. And you can see some of the top ones look pretty good. And we were able to make some of them. One of our top ones here, SW3A, is a cyclic peptide. Again, here's the warhead piece. And we can now put a tag on the end. In this case, we put an alkyne tag, which allows us to now put a fluorophore by click chemistry. And you can see as we titrate into the spike protein, we're getting covalent labeling of that spike protein here. And you can see it starts to label already in the low micromolar range. I should point out that this warhead's extremely slow, so we're finding that we have to incubate for quite a long time, and we're probably going to have to go and soup up this electrophile a little bit to get a much faster covalent binding interaction. But this is a good start. We then tested it in viral infection assays with Ralph Bartenschlager's group at University of Heidelberg, and you can see this is just a control showing inhibition of viral entry. And then when we took our best compound against BA1 Omicron, which is the actual spike protein we used for our panning, we get a reasonably good inhibitor. It's not great, it's 11 micromolar, but it's still inhibiting quite well. And what's really interesting is if we now do the assay on BA5, actually BA1, I should point out, we did BA2, so this is a slight variant. So we saw good activity against BA1, but BA5, which has even more mutations in the spike protein, we get less potent activity here. This is, almost an order of magnitude weaker. So that's suggesting that we're probably getting very specific binding, potentially with BA2. We're working to get the BA2 strain going. Ralph didn't have it running, but we're pushing on that right now. So we're still in the stage now of trying to understand where that binds. We're working with a group that's doing EM structures of the spike protein. We have sent the peptide, and we're hoping to get structures in the near term to see where that cyclic peptide binds. We're trying to use mass spec to map the binding interaction, and that's challenging because the peptide's so large, but we're working on that. Okay, so then the second half, just a little snippet here. I know I only have a few more minutes, but as a second sabbatical, so I learned how to do all the phage stuff in Christian Heinz's lab more than a decade ago. And then this past year, I went to Genentech to do a sabbatical with Christian Cunningham, who does the peptide therapeutics group there, and they make cyclic peptides as inhibitors and therapeutics. And the way they do it is a different platform. It's called mRNA display. I'm not going to have time to go great detail here, but the way it works is you essentially make libraries of mRNA sequences that can be in vitro translated into peptides. And what happens here is that by extending your RNA sequence with a linker and then including a molecule called pyrimycin, if your translation mix has no release factor for your ribosome. The ribosome gets the stop codon and it's making this peptide and it just gets stuck. It sits there. And while it's sitting there, if this puromycin is nearby, it gets taken up by the ribosome and put into the peptide as if it's an amino acid, because this sort of looks like tyrosine. And so now you end up with basically a scenario where you can make libraries of simple DNA oligomers and oligos, and you can take these oligos, make them into mRNA sequences. Then you do a ligation to put the puromycin onto it. And then when you do your translations, what happens is the ribosome ends up making peptides of interest here at this end, and attached to the peptide, covalently attached through this tyrosine or puromycin linkage, you have the actual coding sequence. So it's essentially like making peptides and coding those peptides so you know what they are. So now the other neat thing is, because this is in vitro translation, you can use non-natural amino acids. And you can now make peptides of high diversity, 10 to the 14th sequences, even within a single in vitro translation. You can also make the peptides do cyclizations, so you can get constrained peptides in these in vitro translation mixes. You can protect the RNA after you've made these little peptide hybrids, and now you have this nice set of molecules that can be panned against an affinity binding target. When the target pulls out any of these, you can then just basically heat to remove the cDNA, and then use PCR to amplify the library back up. And so you can do basically what you're doing on Phage, but you're doing it all in vitro. And there's a lot of benefits. You can use qPCR to quantify, and it's a pretty cool technology. The other big advance was this thing called a flexozyme, which is essentially a piece of catalytic RNA that allows you to load whatever amino acid you want onto a tRNA. So now you can take non-natural amino acids, you put this weird ester group on the end of the acid, and that's recognized by the flexozyme, and that allows you to acylate tRNAs, whatever you want. So now you can take a codon table that's got all the natural amino acids that would normally code for a natural linear peptide. You can recodonize it to put whatever you want on those different codon positions. And now you can convert this natural linear peptide in vitro. You can get the ribosome to make these non-natural sequences, including this weird N-terminal capping group. And that reacts with cysteine to form the cyclic peptides. So now you have these really diverse libraries of cyclic peptides containing natural and non-natural amino acids. So what I did at Genentech was to first develop a non-natural amino acid that had a chemically reactive warhead to see if we could build it into peptides using mRNA display. And you can see here, this is this cyanomethyl ester that we need. We have a ketone, and we just do oxime chemistry to make this huge non-natural amino acid. And when I showed this to people at Genentech, they said, I don't know if the ribosome is going to take that as an amino acid. It's huge. But amazingly, it did work. The way you test this is you make an RNA that will express a peptide containing your amino acid that you want to test how well it's incorporated by the ribosome. And at the end, it has this little small bit, which is a peptide that combines with large bit to give you a luminescence signal. So if this peptide gets made and you add the large bit protein, you get a signal. And so you can actually, over time, watch as peptide gets translated in vitro. It's a very cool assay. And you can see phenylalanine here translates really well. If you start to put weird non-natural amino acids, some of them don't express very well at all. So this is kind of the way you benchmark whether your amino acid will get translated. So I did this analysis for mine. You can see here's phenylalanine in blue. That's the natural amino acid. It incorporates really nicely. If we put just a ketone on there, it's no problem. It incorporates. But amazingly, if you put that giant electrophile off of it, it still incorporates. You can see in purple. It's not as good as the natural amino acids, but it's quite good, and in fact good enough to do library screening. And you can confirm this by mass spec. You can see phenylalanine. Here's the natural peptide with phenylalanine. Here's when we incorporated the one with the ketone. It's a slight shift. And here you can see this shift here in purple is this giant natural amino acid now being translated into the peptide. So that was pretty exciting. We could now get the ribosome to make our peptides with the electrophiles hanging off of them. We did many rounds of panning. I did many rounds of panning at Genentech. We targeted FPHB in this case, and we were able to get a number of sequences out here for different ring sizes and different naturals and non-naturals. So it's a long story, but then in my lab, we were synthesizing these. You can make linear peptides with the capping group here that causes the cyclization. We made a series of peptides. And this just shows you real quickly some of the hits we got here. Two of the cyclic peptides that contain the funky amino acid here that we built in by mRNA display. And you can see they inhibit, they're micromolar, low micromolar, which is kind of what we typically get out of our first round panning. We have not optimized this at all. And you can see compared to just the warhead part alone, which is a very weak inhibitor, they're binding pretty well. And then finally, we've been able to show these are in fact covalent inhibitors. We get a nice shift for these binding to the FPHB target. So we now have cyclic peptides that bind. We're in the process of trying to map their specificity and their binding sites. But that's essentially how we're doing it. And it's, I think, a number of different platforms for getting at the same problem here. Anyway, that's really all I had. I wanted to really thank Ralph's group for some of the antiviral work, Genentech for the collaborations, Christian Heinz's group, of course, and then my group as well. So I'll stop there because I think I'm a little bit over.